So tell me which are the biomolecules among all. Option A is DNA, option B is HCl, option C is cyclohexane, and option D is vitamins. So which is the right option? As you can see, HCl is an inorganic acid, cyclohexane is an organic molecule, whereas DNA and vitamins are the biomolecules present in the human body. Yeah, so A and D. So A and D, DNA and vitamins are the biomolecules present in the human body. So this is the right answer. So moving to the next question is, can the nucleobases be synthesized in the laboratory? So nucleobases are basically, what are the nucleobases? So if we will see, nucleobases are the bases which present in the DNA. So DNA is basically composed of nucleobases, sugar and phosphate groups. See, DNA is composed of nucleobases, sugar and phosphate group. And nucleobases are further of two types, purines and pyrimidines. So if you will see, I will show you nucleobases. Yeah, so these are the nucleobases. Nucleobases are the nitrogen containing compounds that form the basic building block of the nucleic acid. So these are the uh, nucleobases. So these two, we are having two rings, and these are the purines. And whatever it is having three bases which is having one ring, these are the pyrimidines. So these are the nucleobases which leads to the formation of DNA. And these, if you will see the structure, then we can analyze that these nucleobases can be synthesized in the laboratory. So yes, these are established. Yes, there are established procedure to synthesize the biomolecules in the laboratory. So yes, nucleobases can be synthesized in the laboratory. So the among the following, the constituent of RNA is. So DNA is full form of DNA is deoxyribonucleic acid, and RNA is RNA is uh, ox, DNA is deoxyribo, RNA is oxyribose nucleic acid. So in this, deoxy is not present, so this is not present. Also, adenine, thymine, cytosine, and guanine. These all are the four nucleobases which are present in the DNA. Wherever in the case of RNA, thymine is replaced with that of the uracil. So in RNA, adenine is present, uracil is present, guanine is present, and cytosine is present. So among all, thymine is present in the RNA, uracil is present, and guanine is present. So B and D is the right answer. So uracil and guanine are the constituents of RNA. However, guanine is present in DNA also, but thymine and deoxyribose are present in DNA only. So these all are the right answers. Okay, so uracil and guanine, these two are the constituents of RNA. However, guanine is present in DNA also, but thymine and deoxyribose, these are the constituents of DNA. So B and D are the only constituents which are present in the RNA. So the fourth question is peptides are primarily composed of, the first option is A, B is nucleobases, C is amino acid and D is lipids. So peptides are basically the, we can say that peptides are proteins. So peptides are composed of amino acids. So amino acid, the polymerization of amino acids lead to the formation of peptides. So C is the right option. Peptides are primarily composed of the amino acids. So moving to the next question, nucleobases are found in, I already told you nucleic acid, DNA and RNA are composed of nucleobases. So nucleobases are found in DNA and RNA. Yeah, so moving to the next question. So the next question is the anomeric carbon in glucose is. So in this, the anomeric carbon is the carbon which is attached to the 2 oxygen atom. If you will see, carbon 2 is attached to one oxygen, 3 is 1, 4 is one oxygen atom. Here that is also attached to one oxygen, whereas carbon 1 is attached to the two oxygen atom. So carbon 1 is the anomeric carbon. So C1 is the anomeric carbon in the glucose. So the complementary strand of the following uh, uh, the complementary strand for the following sequences, we have to tell the complementary. So as we all know, A is adenine, T is thymine, G is guanine, C is cytosine. So A will bind to the thymine, whereas G is bind to the cytosine. So A, here it will come as T, again A, C, G, A, G, T, C, A, T. So the option is T, A, C, T, A, C, T, A, C, G, A, G, T, C, A, T. So option A is the right answer. Okay. So option A is the right answer. A is going to pair with T, thymine, whereas G is going to pair with C. So in a double-stranded DNA, the number of moles of thymine is 0.25, whereas the number of moles of guanine is 0.30. So the number of moles of cytosine and number of moles of adenine respectively. So we should know, in a double-stranded DNA, moles of number of moles of A is equal to number of moles of T, whereas number of moles of G is equal to number of moles of C. So it is asking us thymine is 0.25. So thymine is 0.25. So adenine should also be 0.25. And guanine is 0.30. So the cytosine should be 0.30. So the answer is 0.30 and 0.25. So option C is the right option. 0.30 and 0.25. You can note down. So the next question is the weight of cytosine and adenine molecules are. In the question 2, they have given the number of moles. Number of moles for cytosine is 0.30 and for adenine it's 0.25. That is N. So here they have given molecular weight and we have to tell the weight. So number of moles is equal to given weight upon molecular weight. So you have to calculate this. So you can calculate as number of moles multiplied by molecular weight. So weight of cytosine is equal to number of moles of cytosine is 0.30, molecular weight of cytosine 111. So triple one multiplied by 0.30, 33.3. So there is no need to calculate even second one because cytosine if we will calculate only this is having 33.3. Option A is the correct option. Same you can calculate for adenine also. The next question is ATP is the source of energy in living cells. The nucleobase present in it is. So if you don't know the even the structure of this nucleobases, you should if you will remember that adenosine adenosine tri adenosine triphosphate. So adenosine is the nucleobase present in the ATP. So adenine is the nucleobase present in the ATP. So option D is the right option. You can see. So among the following which will have higher melting temperature. So a double-stranded RNA, double-stranded DNA, or double strand between DNA and RNA. So we all know double-stranded DNA is having the high melting point. So I'm expecting you know that what is the melting temperature. So the melting temperature is the temperature at which 50% of DNA is converted into single-stranded DNA. 50% of double-stranded DNA is converted into single-stranded DNA, or we can say that 50% of double-stranded DNA will be denatured. That is called the melting temperature of DNA. So the number of major grooves in a double-stranded DNA of 1024 base pair is, so if you will see the number of base pair present in one major group is 10 base pair. So if you will see 1024 base pair and number of base pair present in the major group is 10. So we can say that 102.4 are the number of major groups. Yeah. So this is the right answer. So at pH 10 the thymine will exist as, so this you should know. So thymine then, um, yeah. So thymine, the pK of thymine is 10.47. 10.47. That is the pK. pKa, and the first physiological pH of body is 7.4, which means 7.4 is acidic as compared to 10.4. So thymine is present in the acidic medium now, so it means it is protonated. So both the NH will be protonated. So option A is the right option. So thymine will exist as this protonated. So option A is the right answer. So these all were the questions, but you have to see. If you are feeling any doubt, then you can come to me. We will solve this question. So this is all about the assignment. I think these were the two assignments. So I will upload this assignment also, and I will upload this video also in the NPTEL site, and also I will make a folder in which I will upload all the weekly assignments. So you can go through these assignments, and if you are feeling any doubt, then you can come to me. We can solve the doubts. Okay, so now we will meet in the next class. So the next class will also be on Tuesday from 6 to 8 p.m. same timing. So okay, for now, uh, this is done. So we will meet in the next class next Tuesday 6 to 8 p.m. So okay, I'm leaving this class now. We'll meet in the next class. So the right options is, so it is like heparin is obtained from plants and having, this is heparin obtained from animal tissue, um, and this is having two monomeric units, that this is polysaccharide. This is an important biomolecule, anticoagulant in medicinal chemistry, clinically used, conjugation and uronic acid and glucosamine. So the right option is heparin is obtained from the plants. And also peptide can be attached to the free hydroxyl group of cytosine via mite coupling reaction. So these two are the right option. So the second option is, so the second question is, why is chitin converted to chitosan? So first option is chitosan. So chitin is toxic in nature to enhance the solubility, important functional group, or all of the above. So the right option is, if you will see, this is chitin. So chitin is converted to chitosan by deacylation, right? So after deacylation, first of all, instead of this NHC double bond of CH3, this acyl group, there is NH2, which will increase the solubility, and the function group will change so that it will impart the functional group. So B to enhance the solubility and C to impart the functional group. C, B and C are the right options. So the third question is, which of the following statement is true about nanoparticles synthesized from polysaccharides? So it has low toxicity, or the second option is, are biodegradable and biocompatible. Polysaccharides can be used as tox reducing agent and contain functional group on the surface. So if you will see, these are nanoparticles from polysaccharides. So they have low particle size. They are biocompatible. It means that second option is right option. Then they are biodegradable. Then less toxic or non-toxic, has low toxicity. Polysaccharides can be used as a reducing agent. Yeah, and also it can contain functional group on the surface. So all are the true options. So gold nanoparticles. First option is, upon aggregation gives dark pink color. Cannot be used to check single base. Can be used to check single base mutation in DNA. The third option, cannot be used to check immobilizations. So the answer for this is, can be acids with this carbon, all the substituent are attached here. So this is alpha carbon. So alpha carbon, and this is the chiral center. No, this is not the chiral center because all the amino acids have chiral center except glycine. So this is the chiral center because here there is one hydrogen and the other there is no side chain, that is hydrogen only. So all the amino acid except glycine, all are chiral except glycine. So if you see, that is alanine. Alanine is having side chain as methyl group. So this is methyl group is side chain of alanine. And other is tryptophan. Where is tryptophan? This is tryptophan. And other is proline, and one is cysteine. So out of these four amino acid, which amino acid is having R configuration? So you can see there are 21 amino acids. Yeah, there are 21 amino acids and all are having S configuration except one. So first property is all are amino acids with one carbon, which we can tell as alpha carbon, except glycine. All amino acids are chiral except glycine, and all the amino acids whoever are chiral, they are having S configuration except one. The question is you have to tell which amino acids amino acid is having R configuration. Have you understood the question? So can you tell me which amino acids will be having like R configuration? Cysteine. Can you explain, please? Can you explain? Okay, fine. I will tell. So cysteine is having, if we will give according to the priority order, amino will be one. Now, if we will see, this is also carbon. This is also carbon. Now to decide to which we will give second number, we have to see the next atom. Here it is sulfur and here it is oxygen. So which is heavy, oxygen or sulfur? Sulfur only. Sulfur is heavy than oxygen second, and here it is third. So if you will see, going from 1, 2 and 3, if you will see, this is clockwise orientation. So as this is clockwise orientation, so this cysteine is having R configuration. Understood? Yeah. So this cysteine, this is the solution. Option D is the right option. Cysteine is having the R configuration because for cysteine, because of the sulfur atom in its side chain, the side chain has a greater priority than the carbonyl group, leading to the assignment of R configuration rather than the N S configuration. So option D is the right option, that cysteine is having the R configuration. So moving towards the second question. Second question is the number of optically active center for Ella dipeptide is. So first Ella is alanine. You all know Ella is alanine. So as I told you, if we will join two amino acids, or if two amino acids will condense, it will lead to the formation of peptide. If two amino acids are condensing, it leads to the formation of dipeptide. So in one amino acids, I told you there are how many chiral centers? In one amino acids like ALA, there is one carbon center. So if ALA and ALA will combine, it leads to the formation of how many carbon active centers or chiral center? So this will be right. NH2, HCH3, because in alanine there is CH3, C, double bond O, N, H, C, H, CH3, C, O, O, H. Okay, so this is alanine. Alanine, there is a formation of a peptide bond. So in a dipeptide, there is one peptide bond. So alanine, if we will see, there is one carbon atom which is alpha, that is one is the chiral center and this is the second chiral center. So in a dipeptide, the number of optically active center for ala-ala dipeptide is 2. So 2 chiral centers are present in ala-ala dipeptide. So the question third is the amino acids that will exhibit 3 neutralization point when, one minute. Huh. So moving to the next question, third question is the amino acid that will exhibit 3 neutralization points when titrated against NaOH. So you all know, as I told you, in one amino acid, if we will make alanine, there is H, CH3, COOH, and NH2. So in one amino acids, there are one acidic group, one basic group, and one acidic group. So if we will do titration with NaOH, the thing is, um, if we will titrate against NaOH, the thing is there are two neutralization points because of the presence of acidic group and because of the presence of basic group and because of the presence of acidic group. So one will be here and the second will be here. But they are asking for the three neutralization point. So for that you have to see the structure of these amino acids. So glycine is having one group and two group, two neutralization point. Here also two neutralization point. In valine, if you will see, there are only this group and this group, two neutralization point. And here also in isoleucine, leucine, proline, nothing is there, only carbon atoms are there in its side chain. So in all these there is two neutralization points, whereas in the case of phenylalanine, here also if you will see, all are carbon atoms in its side chain, no any other acidic or basic group. So here also there are two neutralization points. In this, if you will see, there are, in above one there is no acidic or basic group, whereas if you will see glutamine, there is having one extra NH2 group, and in asparagine also there is one NH2 group, and in lysine also there is one NH2 group, and arginine also there is a group which is basic. In aspartic acid also there is acidic group, and in glutamic acid. So in option, if you will see, wherever you will find glutamine, asparagine, lysine, arginine, aspartic acid, glutamic acid, in these cases, in these amino acids, you will get the three neutralization points instead of two. So if you will see, in alanine, valine, there are 2 points. In arginine and aspartic acid, aspartic acid and leucine, aspartic acid will be having two neutralization point, whereas leucine, if you will see, it is having two. Valine also and isoleucine have two, whereas in the case of option C, arginine is also having 3 points and aspartic acid will also be having three neutralization points. So the option C is the right option, and the answer will be the acidic and basic side chain. You have to remember, in side chain you have to see the acidic and basic side chain of an amino acid will show one additional neutralization point. So the option C is the right option. Fine. Are you understanding? Yeah. So the option C is the right option. Arginine, it is having basic side chain, and aspartic acid, it is having acidic side chain. So these two are the amino acid which will show three neutralization point. So question fourth is the amino acid that is susceptible to form a dimer is, so lysine, cysteine, methionine, or proline. So what you are expecting? proline. So if you will see the structure of proline, so from where you are expecting the dimer, which bond? First you will see leucine and leucine and lysine, sorry, lysine and methionine. Lysine and methionine. In lysine and methionine, there is no possibility of formation of a dimer, right? But in case of proline also, I don't feel from where you are expecting here. Nitrogen bond. Nitrogen, single bond nitrogen, it is not that much stable, right? Whereas if you see in the case of cysteine, there is SH group. If SH group will be deprotonated, there is a formation of sulfur-sulfur bond, disulfide. So disulfide, if disulfide will form, then it leads to the formation of a dimer. So option B is the right option. If you will see the answer, a dimer of two cysteines linked by disulfide bridge, the presence of sulfhydryl group means disulfide only. The presence of sulfhydryl group means this is sulfhydryl group is SH group. SH group. Where hydrogen can be easily replaced by radical and other group make it possible to form a covalent bond with other molecules. Understood? Yeah. So cysteine is susceptible to form a dimer. So option B is the right option. So you have to tell the correct statement from the following are. So the correct statement from the following are, first is protein folding is a spontaneous process. The option B is presence of beta mercaptoethanol will inhibit protein synthesis. The option C is presence of beta mercaptoethanol will favor protein synthesis. One is inhibition of protein synthesis, C is favor of protein synthesis. And the option D is protein folding requires high energy. So in one option they are telling it is a spontaneous process protein folding. In the other option they are mentioning like it requires high energy. And same for beta mercaptoethanol. In one option they are telling inhibiting protein synthesis and C option will favor protein synthesis. So what you are expecting? Which options are the right options? Yes. But totally A and D are the correct statement. No, no, no. If you will see carefully, A is protein folding is a spontaneous process. Spontaneous process means it doesn't require that much energy. In D, they are telling it require high energy. So both are contradictive statement. So out of two, A and D, one will be correct, and from B and C, one will be correct. So okay, I will tell you. So the protein folding is a spontaneous process. So the protein folding is a spontaneous process under the physiological condition because there is a primary structure of amino acids. When primary structure of amino acids will form a peptide, when these peptide will fold, it leads to the formation of secondary and tertiary structure. This secondary, tertiary, and ternary structure are responsible to do biological activity, not the primary structure. It means to do biological activities, protein has to undergo folding to form the secondary, tertiary, and quaternary structure. It means it doesn't require high energy. It is a spontaneous process only because it has to undergo folding to form quaternary structures, right? So the option A is the right option. Option A is the right option. Sorry, B is wrong. Option A is the right option. Now, presence of beta mercaptoethanol. This is beta mercaptoethanol. If beta mercaptoethanol is there, as it is a reducing agent, this is a redox reducing agent. So, if it is there, it reduces the disulfide bonds. This will reduce the disulfide bond and it leads to the formation of SH and SH. So if two peptides, like if you consider one protein is there, in that protein, if two disulfide bonds are there, then if beta mercaptoethanol is present, so it will reduce the disulfide bond to SH. It means it will break that disulfide bond into SH group, or we can tell it will denature the protein. So it means the presence of beta mercaptoethanol will inhibit the protein synthesis. Means this B is the right option. So A and B is the right option. Whereas this C is wrong, B is wrong. So A and B is the right option. Understood? So this protein folding is a spontaneous process and presence of beta mercaptoethanol will inhibit the protein synthesis. So you understood how presence of beta mercaptoethanol will inhibit the protein synthesis. Okay. So moving to the next question, I think this will be the last question only. No, no. Still we have more. So the PI of alanine, arginine and glutamate will be of the order. So if you will see, alanine is having nothing, but it is having a carbon atom in its side chain, whereas in arginine there is basic side chain. Basic. I am not able to write. Basic side chain, and in glutamate there is acidic side chain. Acidic. So you have to tell me the PI of Ella, arginine, and glutamate will be of the order. Which one is the right option? Yeah, right. So option B, right? So option B is the right option. Arginine is having basic side chain. It is therefore it is having higher pi value. Whereas glutamate is having acidic side chain, it is having lower pi value than alanine. Basically, if alanine is having neither any acidic or basic side chain, so we are keeping alanine as a reference. Arginine is having greater than alanine and glutamate is having less than alanine. So option B is the right option. So the question seventh is denaturation of a protein will disrupt. So first, oh sorry, here the answer is written I think. So okay, fine. So what do you mean by denaturation? Denaturation you all know, right? Denaturation is basically the denature of protein means the protein will the native structure of protein will be disrupted. That is called as denaturation of protein. So as now only in case of beta mercaptoethanol I told you, so the beta mercaptoethanol will denature the protein. It means what structure of protein will be disrupted? Primary, secondary or tertiary? So this is the denaturation. Denaturation or unfolding of a protein disrupts its 3D structure and makes the protein inactive. So urea, beta mercaptoethanol, these compounds are responsible to disrupt the structure of a protein, or we can say that they will denature the protein. So what will happen is it will denature the protein. There is a loss of secondary, tertiary, and quaternary structure in the presence of these compounds. So basically what happen is denaturation of protein will disrupt the secondary structure and tertiary structure. Understood? Yes. So option B and C is the right option. So this was all about your assignment number 2. So if you are having any doubt in the question, you can ask me now only. Anyway, I will upload the slides and recording in the like I will I will record. I recorded this session actually already, but I have to upload this session. I will upload in the YouTube also, and this PowerPoint slides also I will upload. So, are you having any doubt in these questions? Okay. So if you are having any doubt or you are having any doubt in the lecture itself or in the question, you can come to me. We can discuss in the class. If I will be knowing, I will tell you immediately. If not, I will take some time. Maybe next day I will read and tell you. I will clear those doubts. So you have to read these assignments also, and also you have to listen the lectures also properly so that you will be able to solve the questions. Okay, so if you are not having any doubt, so you can leave the class. We will leave. So this only we have prepared for the today. Okay, fine, fine. I think Aisha, you can remove. You can leave the call. Lysine and arginine both are the amino acid having basic side chain, and these are having the positively charged amino acid. It means the polylysine and polyarginine is the right option. So polylysine and polyarginine is the right option. So moving to the next question, dash is used for FMOC deprotection and dash is used for the ester deprotection. The first option is NOH and TFA, second is TFA and NOH, third is piperidine and TFA, and the fourth option is piperidine and the NOH. So if you know, this is the ester is this one. So if you want to deprotect the ester, you need a base. It means we can eliminate first and third option. Now we are left with second and fourth option. So TFA, and out of TFA and piperidine is used for FMOC deprotection and NOH is used for the ester deprotection. So the third option is the right option. So the third question is, which of the following are the peptide coupling agents? So if you will see, the options are DCC, BOP, BOC anhydride and HBTU. So DCC, BOC anhydride is not a peptide coupling agent, but BOC anhydride is used to protect any functional group like NH2 and OH. So it is used to protect the functional group. So out of these four, DCC, BOP, and HBTU, these three are the coupling agents. So if you want to know in the like mechanism and all, you can see the mechanism for these also. So the three options, DCC, BOP, and HBTU, are the peptide coupling agents. So the fourth question is, the peptide coupling agents activates which group in an amino acid? The first option is amine group, second option is carboxylic group, the third option is both amine and carboxylic group. Wait, I will check if some problem is there. Now only I will come to know. I think everything is correct. So I think so. The peptide coupling agent activate the carboxylic group in an amino acid. So the right option is this one, carboxylic group. So the fifth option is, which of the following statement is true for the solid phase peptide synthesis? For the solid phase peptide synthesis is also a synthesis of long amino acid sequence. This is true. Minimized racemization, true. And also it is easy to purify, that is also true. So it means all the three statements are true. Then all of the above. All of the above are true for the solid phase peptide synthesis. So, which peptides are used in the Alzheimer? I think in Alzheimer is a disease which is related to the brain. So, peptide used in the Alzheimer are beta sheet breakers. I feel so. Beta sheet breakers are the peptide which is used in the Alzheimer. In the this assignment, only 6 question was there which I have discussed with you guys. So if you are having any doubt, please join this class. We can discuss your doubts also in this session, so you can see these questions. For the time being, I will mute this my mic. So we will moving towards the question number first. So the following pyrimidine compound can be synthesized from the starting material. So as we all know that this is the pyrimidine compound. Okay, so they are asking that these type of pyrimidine compound, these can be synthesized from the starting material. We have to tell from which starting material we can get these type of pyrimidine based compound. So first is this is A option, B option, C option, and D option. So I will give you some 30 seconds. You can think about that. Yeah, so we will see the solution. So this, so they are asking for these type of pyrimidine based compound. So these are synthesized from this cyanide based this compound and urea. So here they are asking for this substituent at this carbon. So the this will be the right option. Okay. So this will be the right option. So now moving to the next question. So the reaction of guanidine with the beta keto ester would yield. So if guanidine is allowed to react with beta keto ester, then what will be the product? We have to tell this. So so this is like guanidine when if we will see the retrosynthesis. So if um, the guanidine is allowed to react with beta ester, then it leads the two aminopyrimidine derivatives. So when guanidine is allowed to react with beta ester, then it will lead two aminopyrimidine derivatives. So option B is the right option. So moving to the third question, cytosine under enzymatic hydrolysis to produce. So if we will do the hydrolysis, enzymatic hydrolysis, then what cytosine will give? First option is uracil, second is thymine, third is 2-aminouracil, and the fourth option is guanine. So if you will see, this is the cytosine. So if we will add hydrogen in cytosine, then it will give uracil. Then if cytosine undergo enzymatic hydrolysis, it produces uracil. Okay. So the thing is, they have written enzymatic hydrolysis and deamination, because if you will see, it is the hydrolysis also, and also this is enzymatic deaminase because NH2 group is replaced by double bond O. So if they ask cytosine undergo enzymatic hydrolysis or deamination to produce, and the answer will be uracil. You can see. So the fourth question is prebiotic chemistry is the study of, the first is synthesis of nucleobases and amino acids in primordial soup, evolution of biomolecules, protein, DNA interaction, and industrial synthesis of biomolecules. So the prebiotic chemistry is the study of synthesis of nucleobacillus and amino acid and evolution of biomolecules. Option A and B is the right option. So the next question is, according to the Baker's rule, the nucleophilic substitution at Cl negative of the Cl dash of the following will produce. So see this question carefully. For that you should know about the Baker's rule. I don't know why this type of notification is coming. So this is the Baker's rules. So in the Baker's rules, what they are saying is the problem of the glycosylation reaction is controlling the regio and stereochemistry of the reaction. If a 1-2 translation of the two groups C at C1 and C2 dash is desired, like at 1 and 2 position, if translation is less, then Baker rule allows synthetic If the trans relation is required, then Baker rule allow. The oxycarbenium will be stabilized by bridging, forcing an incoming nucleophile to react from the opposite face of the molecule. This rule gives the product shown below. The 1 chloro dearabinose and 1 chloro lyopsols will react with the base to give mainly the A anomers to give the BN hours. So it is telling according to the Baker's rule, the 1 dash and 2 dash position should be trans to one another. So now if you will see this option, already O acetate is here. So if the nucleophile will come like nucleophile C dash will come. So first two option you eliminate because here if you see OS is downward. So if you consider this, so as according to Baker rule that whatever the nucleophile come, it should be trans. So in this option you see they are trans to one another. So option C is the right option. You can see this question. This is the Baker's rule. You can see. Okay, so the next question is the correct statement for solid phase phosphoramidiate chemistry is. So option here it is visible. So it is a biological synthesis of DNA. It is a protocol To manufacture shorter oligonucleotides, it is performed in reflex condition. It required tetrazole as a catalyst. So if you see this cycle, so here first it required tetrazole as a catalyst. If you see, this is a tetrazole. Tetrazole means tetra, four nitrogens. And the other thing is it leads to the formation of dinucleotide. So if you will see this cycle, first is start, then deprotection happened, then activation and coupling in the presence of tetrazole as a catalyst, capping and then oxidation. And if you will see this, it leads to the formation of oligonucleotides. So the option is it required tetrazole as a catalyst, and it is a protocol to manufacture shorter oligonucleotides. So this is the right option. You can see this catalytic cycle also. Now moving, okay, I think these are the six questions involved in your assignment number 4. So you can go through this assignment. I will upload this video, and I will upload the slides too. You can go through this assignment. Okay, so we will wind up the class. So we will meet in the next class. So if you are having some doubt, you can ask me, and I will upload these things into the links. You can go through it. Okay. So, bye for today. We will meet in the next one.